Hello, everyone, and welcome to today's keynote presentation. In advanced cancers, curative therapies must target all cancer cells. Presented by Dr. Krastan Blagoev, Program Director at the National Science Foundation. I am Xavier Gutierrez of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. Our speaker will respond to your questions via email. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're having a problem. This presentation is educational. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Blagoev. I will now turn the presentation over to him.
Hello, my name is Krastan Blagoev, and I would like to thank all of you for watching. And I want to thank also LabRoots for organizing this session and for allowing me to present some of the research that I have been involved in during the past 10 years. My research that I will present today presents some data that we have analyzed over the years from patients that have advanced cancers. And our conclusions are important for understanding how future therapies should be designed. So the simple observation that I would like to share with you is that, very simply, tumors are dysfunctional, dysregulated growing tissue. This has been known for more than 100 years. And also for 100 years, it has been known that these tumors grow even if they initially respond to treatments. In fact, many drugs, of course, first tried in animal models, are delivered to humans because in animals they showed a response of the tumor, that is, the tumors initially decreased. You can see in the graph below, this is a patient who has been now alive for more than 10 years with a castration-resistant prostate cancer. And each line of treatment has reduced the PSA value of this patient, but at one point after a given decrease, the tumor or the PSA value starts to grow again until a given point, and then the patient goes to another treatment and then goes again down, and then again starts to grow after a certain time. And slowly the PSA grows after each treatment a little bit bigger than the previous minimum value. So this is each of these courses are very typical in cancer, and especially the cancers that I will be talking about. These are metastatic cancers. These are advanced cancers. Non-metastatic disease usually is treated locally by surgery or by radiation and chemotherapy, but the ones that I'm mostly interested in are cancers that are metastatic. So before going into the details of our research, I would like to discuss how is normal tissue maintained. So in the past 15 years, we have discovered that tissue that actually renews, like the colonic crypts or the mammary gland or the hair follicles or the skin, is actually hierarchically organized. And at the bottom of the hierarchy stays a small number of self-renewing cells called adult stem cells. And these adult stem cells are non-differentiated cells. They are similar to embryonic stem cells that give rise to the whole body. And they are able to produce any cell in that organ. Organs have complicated structure. For example, the mammary gland has multiple different layers of different cells doing their function. And these cells, these stem cells, are small number of self-renewing cells. We have been able to localize them in colonic crypts, in mammary glands, in hair follicles. Lately, in the past year, we have been able to, people have been able to localize them in the lung. And in general, we believe that renewing tissue has this small compartment of self-renewing cells that give rise to the rest of the cells, which are the functional cells of the organ. Now, these cells, in general, divide infrequently, giving rise to the so-called progenitor cells, depicted here in green. But as they divide and time progresses in our lives, these stem cells, due to natural processes, physical and chemical processes, acquire mutations. And unfortunately, these mutations propagate in time, and at one point, they can become the source of a cancer cell. There are a number of gatekeeping mechanisms or proteins that keep the cells from further dividing once they acquire DNA damage. But often there are mutations also in these gatekeepers, and so unfortunately they don't do their function, and sometimes in the dividing pool of cells, one of those cancer cells is released and then starts to give rise to neoplasm, as we call it, or tumor growth. What are the ways of these stem cells that they can divide? Well, the self-renewing of the stem cells, so in this small graph here, I'm presenting the blue cells will be the stem cells, and the red cells will be the progenitor cells that will not divide indefinitely. But the blue cells, the stem cells, can divide either asymmetrically, that they can divide into one stem cell and one progenitor cell, or they can divide in two stem cells, which we call symmetric division, or in the middle, they can divide into progenitor cells, giving rise to progenitors. In this case, the stem cell disappears from the pool. Now, it is important to notice that in order to maintain organs for a long time, the number of stem cells have to be the same. So the process where a stem cell becomes a progenitor cell, then some stem cell has to divide symmetrically in order to maintain the number of stem cells the same. And of course, if one thinks how many different ways, for example, 4 stem cells can divide, there are 4 different ways that are depicted in this picture. For example, each of them can divide into 1 stem cell and 1 progenitor cell, or some can divide into 2 progenitor cells and some can divide into 2 stem cells, and vice versa. And so this is the 4 possible ways that these stem cells can give rise to the cells of the organ. Okay, so 2 models have been proposed for the evolution and growth of tumors. And the first model, which is based on the hypothesis of the existence of cancer stem cells, is called the hierarchical model. And in that model, there is the stem cell, or small number of stem cells that give rise to all the tumor cells. But that small number of cells divides infrequently and stays about the same in number. In the second model, the clonal evolution model, or also known as the stochastic or random model, in this model, all cells are able to divide, and they randomly divide at random times, and they give rise to the tumor, but there is no special subset of cells that stays small and gives rise to the whole tumor. So these 2 models actually have different predictions for the response to therapies. The hierarchical model, or the cancer stem cell model, has been proposed to explain why most metastatic tumors, after initial decay, start to grow again, and why those tumors become multi-drug resistant. Unfortunately, tumors in the metastatic setting, as we said earlier, become resistant and they start to grow again. Now, the data that we have analyzed uses a criteria called the RECIST criteria, which is used in the United States. And in that case, what is measured using computed tomography, PET or MRI, is the largest length of a given tumor. And In this case, we have actually compared to a more sophisticated three-dimensional reconstructions of tumors, and we have not found very big changes in our growth models. The difference between the three-dimensional reconstructions from computed tomography versus using the RECIST criteria, where we use the largest length of the tumor, is less than 5%. So we've analyzed a number of cancers. We've analyzed breast cancer. Here what is shown are individual patients under treatment. And the points which are given in the graphs, those are points at which the tumor has been assessed, or the total amount of tumor in the body has been assessed. We have also analyzed renal cell carcinoma. These are individual patients that you see both in the breast cancer and the renal cell carcinoma. And after a number of days, as one can see, the tumors first start to decay or decrease in volume. After a certain time, they start to grow. There are various different curves. It's possible that, for example, like in the breast cancer case, the tumor never goes down. That is, this patient was not responsive to the therapy. However, in the third patient, for example, it was responsive and didn't grow much. Different versions of this kind of responses are seen when one analyzes a large number of patients. We also analyzed multiple myeloma, and we also analyzed patients with prostate irresectable cancer under treatment again. In the case of prostate cancer, we have both looked at the volume of the cancer as well as the PSA, which is which reflects actually the three-dimensional volume of the cells that are producing the PSA marker. So the general form of the tumor curve that we observe in this CT or MRI or PET scans, or as well as the PSA, is that initially there is a regression in the tumor, and after a certain time there is growth. Now, it is very important from a mathematical point of view to try to understand how are those regression and growth curves put together, and in the sense of how do they emerge from the populations of cells that are in the tumor, and what kind of mathematical form those curves have, which might be able to tell us something about how cells in the tumor divide, and that might have some implications in terms of treatment and implications for the emergence of drug resistance. Now, one thing that one should realize is that tumors could grow in different ways. We don't really know exactly how they grow. There are different hypotheses. Some hypotheses say that they grow in the bulk, that is all the cells that are dividing, giving rise to the bigger and bigger tumor, are everywhere in the tumor. But there are different hypotheses saying, for example, they might be growing only on the surface, and that in the bulk, that is in the volume of the tumor, they're not growing, and only cells on the surface of the tumor can divide. This, for example, would be case B in the picture presented in this slide. The same is how therapies work. So one would imagine that the therapy could be actually everywhere in the tumor volume, killing cells in the volume, or one could imagine that actually only cells on the surface are being affected by the treatment. For example, in the case of current immunotherapies in pancreatic cancer, some people have seen that the immune cells can penetrate the tumor, and they're mostly acting on the surface of the tumor, and they have proposed that this is one of the mechanisms of resistance of pancreatic cancers to immunotherapies. Well, these kind of different modes of or geometry of sensitive resistant cells would give rise to different actually predictions for the mathematical expressions that describe the change in volume of the given tumor. For example, we have developed different mathematical models, and in these models we combine, for example, cells killed in the bulk of the volume with cells that can be killed that grow on the surface. And so we have a number of combinations. We have 15 models actually that we have tested, and they have different predictions, mathematical predictions for the growth curve and the decay curve during treatment that we observe using, let's say, computed tomography or other imaging methods. So here are the models. I'm not going to go in detail of the formulas. These are not very complicated mathematical equations. These are based on first order kinetics, for those of you who would be interested to know that. And so the different models combined, for example, the first one is a constant number killed and symmetric cell divisions in the bulk. Of course, if the cell divisions are not symmetric, then the growth is different. If they are asymmetric, then the growth goes as a linear function of time. What that means, that just increases linearly. But of course, if they're dividing symmetrically, then in that case, the cells will be growing exponentially. But this will be only when they divide in the volume. If they're growing and dividing symmetrically on the surface of the tumor, for example, in that case, actually, we don't expect exponential growth of the tumor, but something that we call in mathematics a power law. That is, the volume of the tumor goes as some power of time. And for example, one can see the fourth equation, which shows that in this case, the time to the power 3 is how the tumor grows. So what we have gone and tested is all these models on a very, very large set of data that is publicly available that we have taken. These are data from physical trials for cancer trials that actually the pharmaceutical companies or NIH have made available for people to analyze and test their hypothesis and model. So these are clinical trials, again, that we have analyzed in these 4 diseases that I mentioned earlier. And in this case, we have found that the majority, 99% in each cancer, actually obey a very, very simple law. And that is that the tumors decay exponentially, and then they regrow exponentially. This was a very interesting observation because, as you can imagine, if the cells that are driving the tumor growth are dividing exponentially, that is, they're dividing symmetrically, then it seems that the cancer stem cell hypothesis is challenged because, as one can show mathematically, the only way exponential growth can be achieved is really through symmetric cell divisions. Now, in order to get a better understanding of the growth, instead of analyzing individual patients one by one, we have also devised a new procedure called scaling procedure, in which we observe that if we take the patient's data from the point where the tumor starts to grow, at that point, the decay of the tumor is very small volume compared to the one that is growing. So in that case, we can actually simplify the data analysis. And we were able to devise a procedure in which, from the mathematics, we could compare all patients together. And why this is very useful is because, as in any experimental science like physics and chemistry, if we have a lot of data that we can average, then we can actually reduce the noise or the uncertainty in our conclusions. So having that we have, given that we have thousands and thousands of patients, we're wondering how could one compare all of them and average them. Of course, initially it is not possible to average because the initial volume of the tumor inside the body is different from different patients. And also the growth rates and the decay rates are different from different patients. So the only way we could have actually analyzed this data, the one that I described earlier, is patient by patient. But what is interesting that if we ask only the question, are those tumors growing exponentially, linearly, or, as I said, as a power law, that is, the volume again goes as some power of time, then of course one could use our procedure and start the analysis of the data from basically from the lowest point at which the tumor after that starts to grow. Given that procedure, we have analyzed all of the trials that we and all the data that we have, and we observed that on the logarithmic plot, all of the data or majority of the data lies on a straight line. Well, if you remember from high school, if on the logarithmic plot something is linear and stays on a straight line, well, that would mean that the conclusion is that this is actually an exponential function. What does that mean is that as a function of time, the volume or the size of the tumor grows exponentially. And I'm showing here some more data that we analyzed using our scaling procedure or scaling analysis. And all of them, after averages of thousands of patients in each trial or different treatments, show actually a very, very linear plot on a logarithmic scale. Well, in that case, our only conclusion is that these cells must be dividing symmetrically, and that they actually can be described, the tumor kinetics, or how these tumors actually grow in time or decay in time, by a very, very simple model. And in that model, there are two populations, one that is sensitive population and one that is resistant population, or clones. And initially, the sensitive population decays. The resistant clone grows, but initially it decreases because we are killing a lot of cells, and the resistant clone is not growing probably as fast. But after a while, as one can see in the data, the sensitive clone is maybe all of it dead, gone, and then the only one that is left is the resistant clone, which takes over and starts to increase the volume and the mass of the tumor. So the total mass is given just the sum of the cells that are resistant and the sum of the sensitive cells. And what is very important, however, here is that during the course of the treatment, we don't see changes in the growth rate or on the decay rate or the killing rate of the tumor. This is a very important thing in terms of the kinetics, because it really tells us that there is no dramatic changes in the way the cells are dividing in this tumor during the treatment. So again, I would like to emphasize again that these are very advanced cancers. These are metastatic cancers. We have very limited data on non-metastatic cancers, because non-metastatic cancer, as I said, are usually treated surgically, and limited amount of data that we can actually observe a non-metastatic cancer or tumor in the early stages that is growing. In addition, tumors are often, most of the time, really observed when they're quite big, let's say 1 centimeter. 1 centimeter tumor usually has around 1 billion cells. So we really don't know for sure that when the tumor is 1,000 cells or 100,000 cells, what is the way they grow. And this is certainly extremely important for early diagnosis and early treatments, but unfortunately we don't have the data. But in the case of these advanced cancers, we see really exponential growth, leading to the conclusion that a large number of cells must be dividing symmetrically. Now, we re-examined the mathematics of the two models that we discussed, the hierarchical model and the clonal model. And if one just draws a small picture and counts the cells, if we have a number of cells, for example, in this case, we took 6 cells that at random time start to divide. In the first model above, in that model in which one stem cell gives rise to the whole tumor, and then if we take another 5 and randomly start the same process of generating progenitor cells, one sees that on the top graph that actually on the growth is actually linear. This is not a logarithmic plot. This is just a normal plot of the number of cells, and that linearity clearly is not seen in our data, in the data that we have analyzed. On the other hand, if one takes the clonal evolution model, in which stem cells can divide symmetrically and asymmetrically and give rise to progenitor cells. However, these stem cells have to grow, and they have to divide symmetrically and grow in number. So in that case, in the lower graph, one can see that one obtains exponential growth. But the main conclusion of these calculations is that the number of cells, for example, in a billion cells, the number of cells that are dividing symmetrically is of the order of 30 million cells. Certainly 30 million cells are not a small number of cells. And therefore, when we think of therapies and curative therapies, we need to actually target all the cells in the tumor. These 30 million cells, when the tumor grows to several centimeters, are going to be in the billions. And so the main hypothesis or the main idea of the cancer stem cells, that a small number of cells are the source of drug resistance, and we can't cure the cancer because this small number of cells, maybe 1,000 or 10,000 cells, are always there, and we can kill them, seems to be inconsistent with our data that we have analyzed so far. So the model that we are proposing is that actually a large number of cells divide symmetrically. There are probably cells that have progenitor, which don't give rise to exponential growth. But in order for the tumor to grow, a large number of cells have to divide symmetrically. These are the cells, and that is all the cells in the tumor have to be targeted if we want to have really curative therapies. Our research does not exclude the existence of a small number of cells, 10,000 or 1,000 cells, that actually might be completely resistant all the time to the treatments. But at least what we have seen so far, our treatments don't decrease the number of cells below 1 million. And therefore, we have not reached the point at which only few cells have been left, and they give rise to the tumor growth. Now, of course, there are some cases in which cells have disappeared from our imaging modalities. Our imaging modalities can currently can, for example, CT scans can detect 1 million cells. It is possible that in certain cases, actually, it is true that the tumors have disappeared and reappears years later. In this case, there are probably there are dormant cells that are not dividing, they're not giving rise to growth until years later. In that case, these cells, dormant cells, are very interesting and very important to be understood. However, in our setting, in which the tumors actually regrow during treatment, our conclusions are correct. So again, I want to thank you for being here and listening to this talk. And I would like to thank my collaborators, Tito Fojo and Susan Bates at Columbia University, Herbert Levine at Rice University, Wilfred Stein at Hebrew, Julia Wilkerson at NIH, who did a lot of some of the individual patient analysis, and Rumen Yurnov at University of Miami, who helped with the scaling analysis. Thank you very much, and I am looking forward for questions.
Thank you, Dr. Blagoev, for that informative presentation. We will now start the Q&A portion of the webinar, and we'll address some of the most commonly asked questions by our viewers. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. Now let's get started. Our first question is, according to your analysis, do drug-resistant clones exist before treatment, or do they emerge during treatment?
So this is a very interesting question, which is actually at the center of all the debates right now. There is evidence from markers that that drug resistance can emerge from the treatment. Now, what does that mean? Usually, the emergence of treatment, the emergence of resistance in a single cell is very hard to discover, because this is a process that is happening in a single cell. And we usually, when we looked at cells, we have a very small sample compared to all the samples in the tumor. What I can say from our analysis, which again doesn't have the sensitivity of a single cell, is that, as I mentioned earlier, the growth rate and the decay rate of the tumor does not change. From this, I conclude that if drug resistance appears during the treatment as a function of as a result of the treatment, I would expect, if it is in a large number of cells, that we will see a change in the decay and in the growth rates. So we don't see that, but again, our resolution or our sensitivity or our uncertainty is quite big. And so we are not able to exactly answer that question, except, again, the rates are not changing, so this drug resistance cannot occur in a very, very large number of cells. Our analysis of most of the tumors is that at the lowest point, the tumor mass consists of millions of cells. So if millions of cells are drug resistant and they emerge during the treatment, this is inconsistent actually with our data.
Okay, and just one more question. It seems that in your model, the phenotype is fixed. Some cells divide symmetrically, others asymmetrically. But in melanoma, there's evidence that cells can switch their phenotype. Do the conclusions based on your analysis change because of these observations?
So this mechanism was proposed in bacteria in 2004 by Stan Leibler's group at Rockefeller, that phenotype switching is actually the mechanism of antibiotic resistance in bacteria. And I'm aware that in melanoma and has been observed phenotype switching. Our models actually don't say which cells are all the time dividing symmetrically. So there is no contradiction. All we are saying is that at any given time, 30% of the cells are dividing symmetrically. Whether they're the same cells that the previous day divided symmetrically or the previous week, we can't tell that. But all we can assert is that there is a subpopulation of cells, and that subpopulation of cells could be phenotype switching and becoming other kind of cells, while others that are dividing symmetrically become dormant, or they divide asymmetrically. We can't tell that. All we can tell is that there is a large population of cells that are dividing symmetrically.
Well, I would like to once again thank Dr. Blagoev for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. And before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through January 11, 2019. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.